So first of all, I just want to say thanks to Julia and Joe and the team for organising today, but also inviting me to speak and tell you a little bit about some of the research that I do here and on my favourite topic. So we're taking a little bit of a change in stance here. We're not really beneath the surface, maybe on the surface, and we're mostly above ground, so a little bit different. So I've entitled, oops, I don't know what this is doing. So take a breath, and that's because my research really focuses on stomata. So these are the small pores on the leaf, and I'm really interested in the behaviour of those because they're really important. Everybody's had their important things, but stomata are really important for everything we've been talking about today. Photosynthesis, we just talked about the photosynthesis going down from to the exudates down to the roots for the microbial community, water use, and also for keeping cool. So I just want to tell you a little bit about some of the work that we're doing on stomata. So just today I'm going to introduce stomata. I'm going to talk a little bit about some of the morphologies of stomata, and then what triggers them to move, and how does that affect plant performance. And then at the end, I'm just going to give you a brief introduction to 3 research topics that's going on in my lab at the moment. Sorry, there are a couple of graphs, but I'm one of them typical science nerdy people that can't do anything without a graph. Okay, so what are stomata? So stomata are the small pores on the leaf surface. Usually we think about them being on the underside of the leaf. They're on lots of different aerial parts of the plant. But basically these are, it means mouth. Stomata are mouths on the plant, so the plant mouths, hence take a breath. But basically the pores on the leaf, they are surrounded by these 2 specialized cells that we can see here in this image. Maybe not. Those 2 cells that's around the outside, and they open, they allow the carbon dioxide in for photosynthesis, but as a consequence of being open to allow that carbon dioxide in for photosynthesis, they also let water out. And water comes out at a much greater rate than carbon dioxide goes into the plant. So really these are the gatekeepers to that gaseous exchange between the outside environment and the inside environment inside the plant. So really important. And this is what they look like, and I'm going to show you a few pictures. This is from Vicia faba, a typical picture. These are my little pointer doesn't seem to work on here. I don't know if you can see that, but these are the small pores here. These are what we call the guard cells, these specialized pores cells that open and close, allow that pore to open and close on the leaf. But they can also look very different. This is wheat, and this is the guard cells here. They're different shapes. I'm going to come back to that in a moment, and they're in files on these leaves. So I'm going to talk a little bit about the morphology of some of these stomata as well. So just to recap, on the surface, I've said that they're really important for CO2 uptake. That's really important for photosynthesis. If the stomata are closed, and we heard earlier about drought, if the stomata are closed, there's no CO2, there's no photosynthesis. Okay, so really important for those metabolic processes. And of course, that's key to make those sugars and the photosynthates, that's the building blocks for the plants, but also all of these are the processes that we've been hearing about today that links above and below ground. But also that water loss, and I sort of said that in a sort of wasteful way, that water loss. The plant wants to try and maintain water. We're always thinking about the water status of the plant. But that water loss is also really important, of course, because that water loss from the leaf is really important for evaporative cooling, just like us, when we sort of sweat and we lose water, that evaporates, that cools us down. Exactly the same for the plant. And of course, plants are metabolic, have metabolic processes, photosynthesis being one of them, and you have to maintain them at the right temperatures for all the enzymatic reactions to take place. So really important for that evaporative cooling, that water loss. But also, of course, for nutrient uptake. We've heard the fact that the microbial community, the soil, etc., has all these nutrients then needed for all of those plant processes. And they travel up through the plant through this sort of water continuum that goes through from the roots, the soil, the roots, and right through the plant and actually out through the stomata. So stomata are really the gatekeepers. The behaviour of those pores are the gatekeepers to all of these processes. And it really connects above and below ground. So we've talked a lot about below ground and some of the features that's going on there. Stomata are key to this soil-plant air continuum within a plant system and a soil system. So we have, for example, this is a shoot with the root hairs here, these tiny little fibres that increase the surface area to allow the plant to take water up. That then moves through the xylem vessels of the plant, these sort of hollow tubes like straws, and then eventually goes into the leaf through your veins and different vein patterns on leaves, and eventually inside the leaf into here. sorry, I'm bad without a pointer, I like a pointer, into the inside and actually comes out through those stomatal pores that are controlling that water loss through the leaf. And so I just wanted to quickly show you a video. We don't seem to have any sound though.
It brings it great advantages. It allows it to overtop its neighbours so it can get all the sunshine it needs, and it enables it to spread out a huge surface area of leaves, and through their pores it can suck in carbon dioxide from the air. But it also brings considerable problems. As well as carbon dioxide, the leaves need water in order to make food. And water in the leaf can easily evaporate through the pores. Indeed, 90% of the water sucked in by the roots is lost through the surface of the leaves at the top of the tree. But pumping water up here to this height can cause considerable problems. To pump this jet of water 70 feet up in the air here, it takes that huge, big, noisy engine down there. But this tree pumps up about 100 gallons every hour and manages to do so in total silence. How? The answer is to be found in the tree's trunk. The central part of this is wood. Around the outside of this pillar, there are ranks of hair-thin pipes. Those immediately beneath the bark carry the food-laden sap down from the leaves. Farther inside the trunk, there's another set of tubes. These are the ones that carry the water up. They are continuous pipes that extend the whole length of the trunk. As the water evaporates in the leaves above, the long, thin threads of it are pulled up the tubes, into the branches, and ultimately into the leaves themselves. Some of it is used in the food-making process. The rest evaporates through the leaf pores as vapor.
So I love this video because it shows you the root of water going right from the root right up through the plant to outside, and shows that stomatal pore really controlling the amount of water that's coming out of the plants. You can see that's quite a few years old. It was, I think, 1995 that David Attenborough did that first private light for plants. So really, stomata are really controlling that water loss. So water is pulled up through the plants. It's not pushed in through the roots, it's pulled up, and it's the evaporation that actually pulls them long threads of water up through the plant, as well as the nutrients that's within those water. And of course, that's controlled by that little mouth around the pore, around the xylem being like the straw that's coming up through the plant. So I mentioned there's different types. There's lots of different types of stomata. There's two key types that we talk about in terms of research, and we call these kidney-shaped guard cells, which are these ones here. They're typical of the dicots, the non-grass type species, so open or closed. And then we have what we call the dumbbell-shaped guard cells here, that is these ones here in green. And again, like their name says, they look like dumbbells. These are found mostly in the grasses, and of course a lot of our crop plants have these cells. Notice in blue here, there's also what we call subsidiary cells around these guard cells, and some plants have subsidiary cells, some plants don't, and there's lots and lots of different types of those, which I'll show you a slide of in a moment. Sorry, too many clickers. So this is an example of some of the subsidiary cells. They're really supposed to be really important in grasses, which I'll come back to in a moment. But you can see that some plants have no subsidiary cells. A subsidiary cell has to be morphologically distinct to the other pavement or epidermal cells that's on that leaf surface. You can have some that have 2 in sort of weird shapes, but some right up to 10. And of course, don't forget these subsidiary cells are also fully turgid cells, full of water. So when the stomata has to open, it has to overcome that back pressure of those cells around it trying to keep it closed. So again, really important in some of those stomatal movements. And it sort of looks, especially that first image that I showed you of stomata, they look a bit random over the leaf surface, and especially this on the dicots. We see these sort of patterns that don't seem to have any real proper formation. They're not random. It might look random, but there is a pattern, and a specific pattern that is species-specific and environment-specific on all of these different leaves. The grasses are a little bit easier because they tend to be in what we call files and in these rows, so the pattern's a little bit easier to look at. But when you look at any pictures of stomata on a leaf surface, and I'll show you some now, don't think, oh, that's just random, they're just dotted around. They're not just dotted around, there is a specific pattern to all of those that also has implications for the function of the stomata and the plants. So we've been doing some work with Beth Chatto, looking at what we're calling the carbon garden, and we've been looking at different plants and how much carbon they're taking up. Not talking about that today, but maybe another time, guys. But we have taken a few what we call leaf impressions from some of the plants. And so we started in the dry garden and took a few different plants from the dry garden. And of course, you know, the famous, the right plant for the right place. And stomata are maybe a key part of that right plant being in the right place, because there's features in stomata and other parts of the plant that allow them to survive in those conditions. So I haven't got the agave on here, but I'm going to come back to that in a moment. But all of these plants, you can see, have stomata. These are all on the lower surface of the plants. But I hope you can see here, for this plant here, they seem to have a little, like, sort of 2-pairing, but also the stomata are a little bit sunken. They're sort of like in little pits in those leaves, and that's a typical xerophytic adaptation. By putting them into these little pits, it sort of creates a little boundary layer around the sort of opening of the stomata, and that stops that water evaporating so quickly, because it's like you're washing drying, isn't it? If you've got a windy day and everything's moving around and the wind's sort of stirring around the leaves, etc., that water will evaporate much quicker than if it's in its little protected microenvironment. And these are typical xerophytic adaptations that allow plants to grow in certain places, along with other things, and I'll come back to that as well a little bit. We also went to the pond, a different environment, but again, I've said that most stomata and most research is done looking at stomata on the underside of leaves, so underneath the leaves, because they're sort of protected from the bright sunlight as well, that sort of help that evaporative cooling. But of course, if you take something like a pond lily, we're looking at this is the top surface of the leaf here. There are some stomata that seem to be a little bit blocked, but there's a few there on that underside of the leaf. But remember, that's going to be floating in water, so that gaseous exchange is not really going to take place much on the underside as they do in most other plants. But you can see we've got lots of stomatas on the upper surface of the leaf. So there's loads of stomata there, and that gaseous exchange is taking place on the upper surface. And then we've got some lovely pictures from the bird of paradise and some other plants as well. So I don't have time I could just show you loads and loads of pictures of stomata with their different morphologies, but I'll get on. But I did take a few pictures of the favourite fern that's just outside Beth Chatto's house, which is beautiful. You can see here, again, lots and lots of stomata. Seem to be some rows, and then the beautiful veins that you can see there. Interestingly, this plant up here you can see the stomata that's, I don't know if you can with this thing, but there's also seems to be like bubbles on the leaf surface. Again, and that's probably protecting and changing what we call the boundary layer around that and protecting that water evaporation from the plant. So lots of different distinct features. Okay, so what makes them open and close? Well, as you can imagine, there's loads of different internal cues inside the plant, but also external cues to which these stomata or these guard cells respond and change that pore size. So we tend to think that plants open, or stomata open, in response to high light, and there's a spectral influence to that, which I'll come back to. But basically, if you've got high light, you've got the light for photosynthesis, you need the CO2, the carbon dioxide to move in, stomata open to allow that carbon dioxide into the leaf. Decreasing or low CO2 concentrations, the plant measures the CO2 inside the leaf, and again, if the CO2 concentration is low, it means that it's been used for photosynthesis, better open the stomata to get more CO2 into that leaf for photosynthesis. And then when the water status of the plant's okay, so high humidity outside or in well-watered conditions. We heard before that drought will close stomata, as will flooding actually close stomata for different reasons. And then they close in the reverse conditions to try and protect water. So they're continually adjusting and trying to balance that CO2 uptake with water loss. But I just wanted to mention, a lot of my work, I'm looking at light, because that's a very dynamic parameter. We have changes for seasons, years, but also through the day. There's cloud passing, there's leaves fluttering over the top of other leaves, so light's very dynamic. But it's actually, the guard cells are having to look at all of these signals, light, humidity, CO2, pathogens, lots of different signals, and it has to really integrate them and have a hierarchical response. And light is a big driver of some of those hierarchical responses. And I mentioned that light drives these opening. There are exceptions, and I've brought the agave back in from the dry garden. And again, we can see that these stomata are sunken in the agave, which is really nice. But also, agave are what we call calm plants, or have crassulation acid metabolism. And so the stomata don't open with light, and they don't open through the day, but they open at night time. So light doesn't drive them, a completely different signal pathway within those guard cells. And so they open at night time. And when the temperatures are much cooler, there's less evaporative sort of drive and pulling that water out of the plant. They open at night time, they take the CO2 in, and they store it in like the vacuoles, the sort of the water sacs of the plant. They store it as an acid, hence crassulation acid metabolism. So collect that, store it here, and then through the day, they shut the guard cells, shut the stomata, even though the lights come up, and then they re-release that CO2, that carbon dioxide for photosynthesis, back out of those vacuoles, in like I sort of say this to my students, like it's like in a closed jar. So it sort of traps the CO2 in there and it gets fixed with the light energy that's now coming for photosynthesis. So we've got agave. Pineapple is a typical calm plant, as is kalanchoe. And this is why if you have plants in your bedroom at night time, maybe calm plants are the better ones for taking CO2 out the atmosphere rather than the typical C3 or C4 plants. So how do they move? What moves those cells that I've just been talking about, them special guard cells? Well, basically what happens is that solutes, such as sugars and other metabolites, get sort of pumped into these cells, into the guard cells, and then water then follows that just through the osmotic potential of the cells. Water follows it, and basically they just swell up. And they have these sort of radial sort of like belts around them, and that means on the kidney-shaped ones, they sort of just swell up and they're sort of pushing open. And as they push open, they also have to push back against the subsidiary cells that are trying to keep them shut. And that's why nowadays people are thinking them subsidiary cells are acting as reservoirs for both the solutes and the water, and they're just sort of seeing sewn between these two cells. So this is really super interesting for research in the future. And then they close on the reverse. The solutes leave or get metabolized, and then the water leaves and they shut the pore aperture. And so just an example of some of those in real time. This is wheat and oats under the microscope. So we're measuring individual stoma here. Stoma is for single, and you can see them opening and closing. I just thought I'd also mention about the numbers and the size of these. These are microscopic, so they're somewhere between 5 microns and say 70 microns, so 70 microns being 0.07 of a millimeter, so they're very small, so we have to use quite high-powered microscopes to see them. Also, there's a lot of them on the surface of leaves. So for wheat, on the upper surface, a little bit different to most things, on the upper surface can have 60 to 70 per millimeter squared, whereas oak, 500 per millimeter squared on the undersurface of some of the oak leaves. They're packed full of them when you get a picture of oak. French bean, somewhere between 150 and 300 per millimeter squared. Depends on the plant, and it also depends on the environment. The environment will trigger these changes in the number of them. So of course, for us to do research on these, to try and measure even a millimeter squared and look at aperture would be virtually impossible. So we actually measure them through what we call stomatal conductance. So if you hear me talk about stomatal conductance, this is just a measure at a leaf level. We sort of clamp a plant in a chamber. We measure how much water's been lost at a leaf level. So this takes into account both the number that's on the plant, how many stomata are there, and of course, how open or closed are they on the leaf. So it's sort of a measure of the community of stomata that's on that leaf. So why are we doing research in stomata? I've done this since my PhD, which was a few years ago now. It gets frightening when you think about how long it goes. But well, we do this because, of course, and what do we do? Well, we do this because a lot of the work that we do in the plant productivity group at Essex, and I think you'll hear a little bit more about that at the weekend. We've got colleagues that's got some stands going at the weekend as well. We know that we've got 8 billion people on the planet to feed today, but by the year 2050, it's 9 billion. And so our research is really trying to produce crop plants that will grow with increased productivity, but also doing that in the face of climate change. And of course, water is one of the biggest crucial changes that we're seeing with erratic rainfall patterns that we've already heard about today. So really what we're trying to do is trying to produce plants that what we call more water use efficient. Can we get plants that will allow that carbon dioxide into the leaf, keep that photosynthesis going as much as possible, and then but also to save water on the plant, so not letting too much water out. And so if we look at why this is important, why those stomata are important, as I've just mentioned though, but if you look at stomatal conductance on this x-axis here, so this is, you know, the greater the number, the more stomata are open. Against photosynthesis, this measure of CO2 uptake, which is the building blocks for everything of the plant, we can see there's a really strong correlation between those two parameters. Of course, stomata open, CO2 goes in, fixes it for the plant. So not surprising that we've got this relationship. We can also take that a step further, and this is some work back from the 1990s, where you can look at stomatal conductance against a grain yield of wheat. These data were collected in Mexico. And again, the higher the stomatal conductance, the greater the grain yield. And of course, again, more open stomata, more CO2, that gets more photosynthesis. Hopefully that goes into grain yield. But there's two components to this as well. That is one key part. The more open the stomata, the higher the conductance, the more photosynthesis. But also, I mentioned these were taken in Mexico, so the higher the stomatal conductance, the greater the evaporative cooling. And so we've got leaves here with the stomata closed, much hotter than those with the stomata open. These are not wheat leaves, but so part of that was that evaporative cooling. You have to maintain the right temperature for the leaf to be able to photosynthesize, for that to then be produced into grain. So really important, and they're trying to balance that all the time, because of course evaporative cooling means water loss. So what are we doing? I just want to very briefly mention 3 projects that we've got going on in the lab at the moment in trying to see how we could address this. So the first one is what I call speedy stomata. Okay, so I mentioned that they open and close. They do, but some are faster than others. Okay, so if we look at, say, for example, light. So light is very dynamic in the environment. So we've been looking a lot at how dynamic light influences the rapidity of stomata. And so photosynthesis, of course, also responds to light. It needs that light to drive photosynthesis. But as soon as you change the light level, photosynthesis will respond virtually in seconds. It will change the rate of photosynthesis. Stomata are a bit slower. So if we look at a typical sort of experiment that we do, and we look just at this top graph here, we put a plant into a low light environment, so about 100 micromoles. If you think full sunlight on a bright sunny day here in Essex is about 2,000 micromoles, giving it quite a low light level. And we can see that stomatal conductance in the white here and photosynthesis in the black circles is stable. We then switch the light here at this point to 1,000 micromoles, so half full sunlight, really sort of drive that photosynthesis. And you can see that photosynthesis in black immediately jumps up to a certain point, driven by that change in light. And then we have a slow increase in photosynthesis. And that slow increase is as the stomata, sorry, my pointer's not very good, but the stomata in the white circles are slowly opening, and they're opening and allowing the CO2 concentration in for that photosynthesis. So if we look at the graph, and we look at this area in red here, this represents CO2, or photosynthate, that could have been gained if the stomata hadn't been limiting that CO2 when that light changed. So if the stomata had been faster, we had some speedy stomata, we could have gained some of this carbon that it's lost in this dynamic light environment. On the other side, we can see that when the light goes back down to a low light value, the stomata slowly close, a little bit lazy. This is Viscia faba, lazy stomata. Slowly close, and this area underneath, in blue, represents water lost for no carbon gain. Okay, so if we're thinking about trying to balance that carbon gain for water loss, these lazy, slow stomata are losing water but not gaining any carbon. So can we speed that up? And so that's part of what we're looking at, is trying to speed that up, looking at different anatomies and morphologies, but as well as some of the metabolites and the processes that go on in the guard cells. And so if we're trying to look and screen populations to see if they've got speedy stomata, we can use a few cool techniques to do that. And this is an example where we use thermography. And so this is a wheat panel that we've been looking at in the lab where we've used thermography. We've changed the light, and then we look at thermography to see how the stomata are behaving, because, of course, leaf temperature is driven by how open the stomata are, and so we can therefore see if there's differences in the plant. And so what you're seeing here is real-time conductance, spatially and temporally, with a step change in light. And I think what's amazing about this is you can also see that spatial heterogeneity that you see in the leaves. And that's because you've got these spatial patterns of stomata that result in spatial heterogeneity of behaviour in them leaves. But this is a really useful screening tool for us to be able to get through lots and lots of populations to look at what might have speedy stomata. Okay, so the second project I want to tell you about is my blue light project. I don't have an answer for this. This has been going on about 10 years, but one of my favourite projects. And basically, I mentioned before that plants respond to light. So plants like us see the visible spectra. So the visible spectra here, sort of blue to red, that 400 to 700 nanometers, exactly the same as what we see. And blue and red are really used for photosynthesis and dry photosynthesis, a little bit of greens and yellows, but they're not absorbed so much as we know. So when you learn about stomata and the response to light in a textbook, you'll see something like these graphs here, is that, you put on light, and we're going to talk about just red light here, not blue, but red light being sort of every bit of light that drives photosynthesis, you see this sort of typical opening response. So on this graph, we've put, say, 1,000 micromoles of light on, we've sort of saturated photosynthesis, we've saturated stomata. So this red light response is sort of that link between photosynthesis and stomata, that correlation I showed you before. However, if we then put 20 micromoles of blue light on, so we've got 1,000 in red here, and we put this tiny, tiny amount of blue light on here, really, really small, it's not going to drive any photosynthesis, we can double stomatal conductance in these plants, in some plants. And this is what you learn when you're at school or in the textbooks. That blue light drives this sort of fast opening of stomata, 20 times more effective at opening stomata, etc. And so we started to look at this, and what we found is that not all plants have this blue light response. So it's not standard textbook blue light response. And some have it a lot more than others. So we looked at two different plants, one that sort of has a blue light response, and that is wheat. So wheat, one of our key crops, and tobacco, which is one of our model crop species. Tobacco doesn't really have a blue light response. So if we look at plants, stomatal conductance, again, how open these stomata are, and we've given them this sort of diurnal light pattern. We can see here the stomatal conductance in tobacco, there's not really any differences with the stomata responding to that blue and red light. So sorry, red light or red with a tiny bit of blue in it. And you can see that the photosynthesis follows the same pattern again on this side. There's no real difference. It's not affecting photosynthesis. If we look at wheat here, the paragon wheat, we can see that blue light drives this massive stomatal opening. Okay, so you've got this much higher stomatal opening, much more water being lost from these plants, but very little effect on photosynthesis on that right-hand side. So I thought, should we just try and knock this out in the plant? Should we try and get rid of it? And that's what we've been trying to do, is trying to get rid of this out of the plant. And luckily, we've got one gene that we can do, and we're trying to knock that gene out that's in the guard cells. I'm going to skip this just a little bit for time. And then the final thing I want to talk about is just the fact that, and so I should say, we've not managed this in wheat yet. It's really annoying. I've got wheat plants and they're not quite there. But so the other thing is non-foliar photosynthesis. I've mentioned that we always look at stomata on the leaves. Of course, they're also found on lots of different parts of the plant. They're found on the stems, they're found on the ears of wheat, etc. And so we've been really interested in looking at non-foliar tissue. And so you can see that if we look at the ears or the spikelet of wheat, these awns here have lots of stomata. There's about 300 millimeters per millimeter square on these awns. What are they doing? We know that these are green. There's probably some photosynthesis going on. But are the stomata functional, and what are they doing on the plant? So we've actually looked at that, and we can see that they are functional. We've done, again, this change in light. There is a bit of photosynthesis going on. These numbers are quite low here. Don't worry about the different lines, but just see the pattern. But the stomata are functional. They are opening in response to light. But what are they doing? Are they important for cooling that ear? Are they really important for the translocation of photosynthate up to that ear for grain yield? And that's something we're trying to now explore in a little bit more detail. And again, to do that, we've got thermography that we can do that. We can screen some of these responses in some of our populations of barley and in terms of wheat. And we've got an example here where we've got a plant here that has more stomata. This plant has much more stomata in the ears, and you can see that it has about 20% more stomata in the ears here, and that leads to a 1 degree difference in the ear. And we know that fertilization is very temperature sensitive. We know that we need to get the photosynthate up here. So again, is this something that we can look at to make plants more water sufficient? We know we're losing a lot of water from these ears. The numbers on that graph said that was quite a lot that was leaving from them. So what can we do to look at that? And I'm just about out of time, but before I finish, I just want to mention the fact that we have looked at a few different pods from Beth Chatto's garden. We looked at the Agapanthus pod. We've got some stomata on the leaves, unfortunately nothing on the pods. Interestingly, the Gunnera, which is a really interesting surface of the Gunnera with these massive leaves underneath, etc. We did find them on the flower stalks of Gunnera. So you can see stomata that look exactly the same as the leaves. What are they doing on there, and why are they important? So again, a lot of people have not really looked at these yet at the minute. So I want to finish there, say thanks to my lab, who actually do all the work. But also I wanted to finish with, we've done a time-lapse for Beth Chatto's, looking at the Gunnera grown, and I hope you can see some of this grown over time that we've set going at the beginning of this year as part of our carbon garden work with Beth Chatto. So thank you very much.
Thanks, Tracy.